Good morning. How was everybody? How was your morning session? I hope you enjoyed the engaging and networking time. And we want to thank our gracious sponsor, Intercept. Is there anybody from Intercept that's here? I don't see anybody, but if you do run into somebody from Intercept, make sure you thank them. We want to welcome you to our first breakout session of the day. This is track 3, Patients as Drivers in Early Drug Development. I'm Alan Gilstrap, and I will be your breakout host for this morning's session. I would like to also welcome those of you joining us remotely through our live stream, and would like to remind everyone joining us to download the mobile app if you have not. There you can find agendas. You can, if you don't feel comfortable at the end of, during the Q&A time, you can ask questions on the mobile app so you can remain anonymous and you don't have to get up in front of everybody. So you have time to download the app while, before we get to the Q&A. Today's session, Early Drug Development, What Patients Bring. We will hear from experts, advocates, on the different stages of drug development process and how and where patients can have a role. Today we will be hearing from Sharon King, President of Taylor's Tale, Steve Gray, Associate Professor, Department of Pediatrics at the University of Texas Southwestern Medical Center, and Laura King Edwards, co-founder of Taylor's Tale. Let's welcome them. It's all yours.
All right. Good morning, everyone. I'm Sharon, and it's so nice to see so many of you in the audience. My rare disease journey started before Global Genes was even organized, and to have this kind of organization that brings everyone together, gives us a chance to share our stories, our experiences, our hopes. But it has made such a difference. Taylor's Tale was an early part of the Foundation Alliance, and again, a place to coalesce, to ask questions, to share insight. So thank you, Global Genes, for all you do for this community. And we want to we want to continue to share our story as well and to help others. But good point that we started, when we started, we feel like it was 12 years ago, almost 13 now. And we feel like things have changed so much since we actually moved into the rare disease space. But most importantly, we're not alone. We're all together in this. When we talk with one another, we realize that our stories all feel the same as we move forward. So thank you for being here, audience. Oh, there we are. Okay. So when I see this verse, I think so much. Those of us in the rare disease community use running as an analogy so often for what we do, and it is because time is so urgent for us. It is a race, and we are racing against time. And you think about running a race. So I'm not a runner, but I gave birth to 3 runners. And I've learned a lot about running through them. I know that you start out, there's a lot of training to get ready. You do your homework. You have to understand your course, where you're going, where are the pitfalls, the potholes, the hurdles. What do you need to know to make it to the finish line? Are you running a relay race? Who's with you? Who's on your team? How are you going to work with them? How are you going to continue to hand off the baton and make those smooth transitions? And importantly, do you believe that you can actually get to end goal? Can you cross the finish line? And so, yes, it feels very much like a race and you also will have to persevere. Come on. There we go. So I'm not being a runner. I like to look at it in a little more simply, and I call it the ABCs of hope. First off is hope requires action. If you're going to sit back and wait, you've gotten your diagnosis. If you sit back and wait for someone else to fix it for you in the rare disease space, you might be waiting a long time. And I always come back to the fact that time is so important. You're going to have to believe, can you really accomplish your goals? Have you done your homework? Do you know what it's going to take to get to the finish line? It's going to take a lot of commitment. Taking this path, this journey, is a 24/7, 365 commitment. If you think that you're going to get it done 3 hours on a Saturday afternoon, you may not get to your finish line. So go into this knowing that you're committed to the end goal. And then, most importantly again, do you have the courage to see it through? Because you're going to come up on a lot of roadblocks. There will be those potholes that you're going to come close to breaking an ankle. You have to have the courage and the commitment to move forward. It's beastly, but it can be done. You have to believe. So, our story begins with a little girl named Taylor. She is our why. We were a happy family of 5. Taylor was a bonus. Laura was the proud owner of a driver's license when Taylor was born, and Brother Stephen was in middle school. And Taylor, of course, was the supreme ruler of the family, or at least she thought so. But, as I think back, there were clues early on that there was a monster in my daughter's genes that was threatening to steal our happiness. And in first grade, everything seemed to be very normal. But in first grade, I was volunteering at her school, and her teacher came to me and said, we've done some standardized language testing, and her scores are all over the place, and they don't seem to be what we would have expected from her kindergarten experience. We agreed to retest her, and at retesting, things looked just fine. In second grade, we took her trick-or-treating, and literally her friends were laughing at her because she was falling up steps. She was tripping over plants and shrubs and falling into shrubs as if she couldn't see. And additionally, we noticed that when she was watching television, she would turn her face to the side as if she had an impaired visual field. So we decided to take her to a pediatric ophthalmologist who announced to us that everything was just fine. But in fact, in reality, things were not fine at all. It took another 6 months, and in that, we know that we were fortunate for things to move as quickly as they did. It took another 6 months to get a diagnosis of retinitis pigmentosa. And we thought, okay, we can deal with this. We'll find the specialist. We're going to get involved with the Foundation for Fighting Blindness. We're going to raise money, and we're going after a treatment, and we're going to find something for this. A month later, we were invited to a geneticist's office where we got the real diagnosis. He told us that our sweet 7-year-old, had CLN1 disease, which is commonly called Batten disease. We were told that she would continue to lose her vision, that she would have seizures, she would lose her ability to walk, to talk, to swallow, and when the monster had stolen almost everything it could from her, it would then steal her life. Well, I'm thinking about my sweet 7-year-old, and I'm thinking, no way. Batten disease will not win without a fight. I didn't have any background to figure out where to go, was there a way to develop a treatment? And the geneticist had just informed me, no treatment, no cure, go home, make happy memories while you still can, handed me a stack of papers from the internet describing Batten disease and sent us on our way. What I did know was I knew community service. I've been involved working in Charlotte community activities for many, many years. So working with Laura, we pulled together family and friends, and we asked them to help us find a treatment, something that was hopeful for Taylor and other kids like her.
Just real quickly to add to that. My mic, everyone can hear me? Good morning. Just wanted to add to that. I think that certainly looking out in this room, a lot of you are patient advocates, family members, affected patients who maybe are thinking, what do I have to bring to the table? I don't have a background in science. I don't have even the fundraising experience. What can I do? But I think that one of the things we hope you can take away from this session is that we all have something to bring to the table, and part of getting started is understanding what that special sauce is that you have that you can bring to the table, and then how do you build that team around you to make sure that you put all of the pieces together.
Yeah, and that's what I had. I had community experience. I knew how to organize. And then we found out, and we also understood that you have to do your homework. You have to know what's out there. What do you have to work with? Where do you go? You've got to ask the questions. There were lots of long nights on my laptop after everyone else had gone to bed, reading, asking for people to share papers, consulting with scientists, going to scientific conferences. Less than 6 months after Taylor's diagnosis, we attended the World Symposium on Lysosomal Storage Diseases. And that was my first real taste of going out into a scientific conference. There were lots of terms, words that I didn't understand, but I figured them out. I did my homework. And we also started with CLN1 disease. We had a couple of things going for us. The gene had been identified, and there was a mouse model. We started fundraising. Within 6 months of diagnosis, we started fundraising, and we, with our first fundraiser, we were able to raise enough money to start a project with enzyme replacement therapy. There were other projects along the way, but in the early days, our focus was ERT. We had multiple fundraising events before we decided to go after a nonprofit designation. Come on. There we go. To turn hope into, so our whole goal was raise money, figure out the best places to put it. We didn't talk a lot about what all of you know is so important now. Registries, natural history studies, understanding the regulatory process. In the beginning, we just continued to think, if we can raise enough money to fund projects, we'll get to where we need to be. When you're a mom raging against rare disease and time is so critical, you get really impatient. Every year we continue to fund projects, in particular enzyme replacement therapy. But I was getting really impatient. I was watching the days tick off the calendar and watching my daughter continue to become more and more apparent that the disease was progressing. So I was at a workshop in Bethesda for CLN1, CLN2 disease, and a young scientist went to the podium and presented a project in his lab, a small pediatric neuromuscular rare disease called giant axonal neuropathy, or GAN. This project, and he shared that it could have some promise for CLN1. Some of you could be modeled for CLN1 or CLN2. I was mesmerized. I thought, this is where we need to go. I'm I want to learn more about gene therapy. So when I walked out of that conference room, I was 80% sure I needed to talk more with Steve Gray, but I needed that 20% due diligence. I had to do my homework, I had to learn more about gene therapy, I needed to understand what we were getting into. I went back to the Taylor's Tale board. They were not quite as enthusiastic because they had invested a significant amount of funding and time into moving forward another project. They were concerned that if we deviated, that we would lose focus. But I was absolutely sure we needed to move towards a project in gene therapy.
Yeah, I'm just going to jump in.
Jump
real
right
quick,
in.
Too. Yeah, so this is one of the things. There will be an overall theme about staying focused, and once you have something that looks promising, to try to avoid the distractions and stay focused on your goal and on moving it forward. But I think this is a case where I think they had a promising therapy, a promising avenue for a therapy, but Sharon stayed open-minded to consider emerging technologies and really keep an eye out for other things that are going on. And it was really trying to benefit, keep in mind the perspective of, when Sharon was doing all of this, it's a much different time now. There's a lot of new innovative treatments that are moving forward for rare diseases. And this was a case where, trying to come in at the beginning of things and taking risks, to move something forward. And, this odd combination of staying focused but also being open-minded to other promising technologies.
Yeah, and gene therapy was just moving into this new state of...
and also trying to build on successes of others and then trying to apply that and trying to see things that are sort of platform approaches that can span across multiple diseases and kind of copy and paste efforts and build on other successes.
Right.
Yeah, I would almost term it as measured risk-taking. I mean, you have to have the courage to take risks when you're in this business of fighting rare disease, and no matter how desperate you are, how personally it hits you and your family. And I say measured risk-taking because all of that upfront work of doing the research on your own, getting to know the people in this field, becoming an expert, becoming a citizen scientist if you're not actually a scientist, all of those things are going to help you and informing you in taking those risks that, they might not pan out every time, but they could. So those educated, measured risks are going to be really important in moving your fight forward.
The way we finally move things forward. So the GAN study was moved forward by a group called Hannah's Hope Fund, led by warrior mom and advocate Lori Sames. I needed to get a study going with Steve, and I needed for the Taylor's Tale board to believe along with me. So I invited Lori to a board meeting. And any of you who know Lori, she has very much a get on board or get out of the way, we've got work to do kind of attitude. And she also makes it her business to know her science. So Lori came to the meeting. How could they resist? The board moved forward. We decided we would get involved with a gene therapy study. The other problem was the funding piece of it. We had made a huge commitment to other projects, and we didn't believe we had enough money to get in to make that significant first push. So we were able, over the course of about the next 6 months, to join on with an international funding partnership to actually get that initial 3-year study going in the lab. So I guess what you need to pull from that is, if you've done your homework and you believe enough in this study that you want to get involved and move forward, you may have to be innovative in more than the drug therapy. You may have to be innovative in how you think about actually accomplishing your goal. Would I have ever thought that we would have joined on with this international partnership? No, but it worked, and it allowed us to move the work forward. And I'm
And
gonna,
I think that's about being creative, too, in how you approach this fight. You know, there's an old saying, I know you've all heard this before, but where one door closes, there may be another opens, or there may, maybe there's not another door, but maybe there's a window somewhere. And so I think that was a key for us in this, was understanding that no didn't have to be the final answer if we didn't have all of those pieces in place right then to move this forward. We had to get a little creative, but we were able to make it happen.
Let's see.
yeah, there you go, yeah. So in my work in convincing the board, I took board members up to Steve's lab to actually see what was happening and to, get that feel. And then, of course, after we made the commitment, we hosted a breakfast in the community, had about 100 people show up to actually announce that we were actually going to move this forward. And so the picture with Laura and Steve comes from that. So,
That was on, for context, that picture on the right was Rare Disease Day in 2013.
yeah. I was not there, I was actually in Washington.
All right, so the point of this really isn't the science, but I wanted to go through just a little bit and not get too technical, just to really illustrate a couple of points. And I mean, one is this point that I've already stated is benefiting from other people's success, seeing what's happening in other diseases, and then trying to take their success and apply it, try to transfer it to how it can help your disease. Reinventing the wheel is tough. And so I'm talking about gene therapy because it's an emerging technology that's showing a lot of promise really across hundreds of rare diseases. And it's not a one-size-fits-all. In most cases, it's not a cure. But conceptually, most of the rare diseases that we're talking about, I think 80% of them are genetic. So if you identify the defective gene, as most of you have, then the idea is to just package the working copy of that gene in some kind of a carrier, like a virus, and then use that to shuttle it back into the body. One-time injection, potentially lifelong treatment. So it's really changing the paradigm of medicine. But it's an emerging, exciting technology. It's still early days. But I kind of put like how gene therapy works. How does it really work? Really work, means you have this idea, we can replace the gene and we can fix things. The devil's in the details there. So, first you have to have a model of the disease. These are usually mice. You have a sick mouse, you make the mouse healthy. Sounds good. And then you talk to the FDA. Then you have to do all kinds of safety studies. And then you have to get permission to start testing this in patients. And one of the things that we talk about, if we can just get into that phase 1 trial, if we can just start treating patients, that's the start of the experiment, a phase 1 trial. That's not crossing the finish line. That's getting to the start. So it's a goal and it's a significant milestone, but it's not the finish line. And of course, this is okay, how this really works to start testing a new treatment. But the reason why this takes so long, and I'm not telling anybody anything they don't know, is because each one of these steps has funding or needs significant funding. And I'm an academic coming from the academic world. If we were going to do this through, say, federal grants, each one of those 3 funding steps is a separate 5-year grant that we may or may not get funded. So you can see where, if this is, taking a standard academic track through federal funding, this might be a 15, 20-year process.
And I think, Steve, that underlines, too, one of the points you made early on. I mean, we are the rare disease community, and there are at least 7,000 different rare diseases that any one of us in this room is facing. And one of the issues that I think this community has had historically is that we've kind of tried to work on our own little silos and figure out a disease over here, and then figure out a disease over here, and meanwhile not building on the knowledge that each of us has gained in our little silo. And so when Steve puts up here on the slide that you have funding and funding and funding, and it gets really expensive, and you have these families who are facing diseases, and that's hard on its own, how are we going to raise millions of dollars and then somebody else has to turn around and do it again? And so I think from a science perspective and funding perspective, It's really important that we start to break down those silos and understand what can we learn from people who've done it before, even if the disease is different. And you start to gain economies of scale. So that's really important as well.
And that's what we love, that from the Taylor's Tale, when Taylor's Tale went after this, it was that we could potentially have, produce this promise for CLN1 disease. But one of the things we really loved about it was the fact that we hoped that we could model GAN and that someone else would come in and model us, and that actually has happened.
So I'll touch on that a little bit. And, as far as the science goes, this is where we're using something called AAV, adenoassociated virus. This is becoming a very prominent way to deliver genes in the gene therapy community. There's 2 FDA-approved drugs now that are for SMA and Leber's congenital amaurosis that are using this virus. But we were one of the labs around the world that was really identifying ways to use this virus to deliver genes broadly across the brain. And this is just showing like a slice of a mouse brain, and all the dark there is where a gene is being expressed that we delivered. So you'll notice it's not every cell, but it's a lot. And so we're looking at some diseases where it could potentially be a cure, but most diseases is the treatment. So, I love doing science for the sake of curiosity. That's why I became a scientist and understanding things. But the reason that I'm really here and talking today and doing what I do every day is, I can say it started with this girl here, Hannah Sames, who got diagnosed with giant axonal neuropathy. And similar story that everybody knows in this room, the doctors told them exactly what was going to happen to her and that she would die at about 20 years old, and that nobody could do anything about it and enjoy the time that they had with her. And her parents, Lori and Matt Sames, created this foundation, Hannah's Hope Fund. Within 5 months, they put on the first ever worldwide symposium on giant axonal neuropathy. I attended that, and 2 months later, I had a pretty significant grant to try to develop gene therapy at a time when the technology to do what they needed didn't exist. I could spend 2 hours talking about this story, about all the amazing things that had happened. But let's see if I can get this to advance. I'll just sum up a lot in one slide where, in 3 years, we basically developed this approach to deliver a gene across the brain. We got good lab results, which means that we treated cells, we treated mice, and then we went to the FDA in January 2012, say, we want to try to deliver a gene in these kids in a way that nobody has ever done before for any disease. But we think it's really important because these kids don't have any other option. And I'd like to think that we presented everything in a very good way so the FDA understood it. But the short of it is they got it. And I'm not going to badmouth the FDA at all because they were incredibly helpful to our effort. And the basic gist of it is they said, we support this. This looks good. Just do it responsibly. Now, do it responsibly means following a lot of federal regulations and all this stuff that I didn't really know how to do. But then we moved into doing formal toxicology studies, designing clinical trial, going through this alphabet soup of regulatory agencies. Our application to start the clinical trial was it was something like 2,000 pages, but we did it. And in 2015, this amazing thing happened that we started a phase 1 clinical trial. And I'll say that this was pretty much 100% funded by Hannah's Hope up to the clinical trial. And that was something, the family they told me from day one, you move the science forward. We'll figure out how to get you the money when you need it. And it was a relationship of trust and a relationship of just, we were all driving towards the same thing with the idea of like, we just can't fail. Like, it's a cliche, failure is not an option, but I mean, if we fail, then these kids that I came to know and love would die. Simple as that. And so every time we hit a wall, every time we hit a roadblock, we'd figure out something else. So So anyway, this was, a milestone in gene therapy was, May of 2015. We did the first gene transfer, by a spinal tap for any disease. And it happened for this rare disease that to this day we know of less than 100 kids in the world that have this disease. But it happened for this disease, and it happened because you had a patient advocate, a mom and a dad, that made it happen, that didn't accept that there wasn't anything available.
And Steve, how many, sorry. I was just hoping real quickly Steve could share with the audience about how many diseases you're working on now that in some way, shape, or form are connected to the work you've done with GAN.
Well, yeah, so that's when I talk about a win for one is a win for many. Let's see. I think this might be the next slide. Okay, so look, we're not the only people doing work with AAV vectors, and we're not the only group targeting the brain, but this is just a kind of a short list here where you had a clinical trial for spinal muscular atrophy that started in 2014. That's now an FDA-approved drug. Our GAN trial was the first intrathecal trial. This is all with the same version of virus. So all of these all these treatment approaches they're basically using the same technology, the same approach. And you see one in 2014, one in 2015, two in 2016, two in 2017, and now we've got like a bunch starting in 2019, and it's really exploding. This isn't a comprehensive list. I don't know every trial that's being run, but it's really, and these are from different groups. So there's communication, there's other labs are picking up the same technology, other disease advocates recognizing the technology, saying, hey, can this work for my disease? Find a scientist, get it going. So, my perspective, and everybody has a different perspective depending on where they're coming from. My perspective, I'm an academic. I run a research lab. I'm a basic scientist. I'm not a physician. But my perspective working with a lot of different families and being really at the heart of the science is the best thing I can say as far as magic ingredients are link scientists and families. There's different things that you have to have compassion. I think if the scientist that's involved is just in it for intellectual curiosity and they don't want to get to know the patients, they don't want to understand they're just not going to have that same drive. You can have compassion, but if you don't have know-how, you can have every intention in the world, but if you don't know how to do it, it's not going to happen. So that's really linking the patients and the families. I think the accountability, if you're working with a scientist, don't get your one-year written report on an annual basis. Like, keep in communication with them, know what's going on. Make sure that they know your kids. And these things don't get paid for by themselves. People don't have magical pots of money. So it's absolutely horrendous how this has to work, that somebody has a really sick child that they're trying to take care of, and then they have to understand the science, and they have to raise a lot of money, and they have to organize meetings. Like, it's the most unfair system in the world, but I'll tell you, if it doesn't happen that way, it's not going to happen.
And I will add to that as well. One of the things that's really exciting to me, and hopefully for you, no matter where you are in your journey, is that one of the slides Steve put up on the screen earlier was kind of showing the timeline for the GAN work that Hannah's Hope Fund funded, really drove forward. And if you think about the time frame for that, I think it was about 6 years and millions of dollars that this little organization for this little disease was able to raise. And I will say that Lori Sames and her group, that story is not a typical story, and it is not going to happen. That's not the way it's going to happen every time. But we've been able to benefit from the economies of scale. And what I mean by that is being able to, again, build on past successes. And so in the work that we were associated with Steve on CLN1 disease or Batten disease, it didn't take as long, and it didn't, frankly, take as much money, because we were able to lean on some of the experiences and understandings that were developed previously. So that's promising to me, knowing how many diseases we need to tackle.
Yeah, and also I think the landscape has changed for a lot of these rare disease treatments where, I think when we started, it would have been unthinkable that a company would invest in something as rare as CLN1 Batten disease, but it happened. And it took the foundation really providing the seed money to do the really good science and to show that this had a really good chance of helping patients, to de-risk it. That's the word, right? De-risk it enough that a company was willing to invest in it. So, the summary before I kind of hand over to Laura is, just a couple points I want to make sure that I touched on is, gene therapy is creating platform technologies. It's making treatments available for a lot of things. You have antisense oligonucleotides, which is like a huge emerging treatment that's going across multiple diseases. You have drug screening. You have lots of different approaches now that are starting to show successes in the rare disease. So I think you need to understand the biology of your disease enough to look and say, well, what are other diseases that face the same issues, the same problems from a treatment perspective, and try to see where someone else has had a success and how it can apply, whether that applies to your disease or not. But I'd say stay away from the glitz and the glamour. There's a lot of really, there's a lot of stuff that it's really cutting edge. It's like it's brand new technology. It seems like this is going to be the thing. That was gene therapy 20 years ago. And it's really taken like 15 years to go from the promise to actually producing. And then I want to say stay focused on goals and the danger of rabbit holes. This is particularly like, scientists love to understand everything. And I think we succeeded because we always focused on the goal of we have to develop a treatment. And everything that we did fed into, is this going to help? Is this going to lead to a treatment? Or is it just going to answer a scientific question? And so we stayed very focused.
After Sharon met Steve at a meeting in Bethesda in 2011, I think late 2011, she came home and couldn't stop talking about this young guy who had all these great ideas. And my first question, having not been at the meeting, was, well, what about XYZ that we're already doing? And one of the first things out of her mouth was, yes, but it's translational research. She kept really emphasizing that word translational. He has that tunnel vision can be dangerous, but tunnel vision in the right areas can be really powerful. And I would say Steve has that tunnel vision of everything we do needs to be very focused and very directed toward how, getting that treatment into kids. Or whatever patient community that we're focused on. Publishing a paper is great, getting that additional grant's great, but why are we doing this?
So I think with that, I'll maybe pass it off to Laura, and we'll also have a lot of time for questions.
All right, so Taylor's Tale uses running analogies a lot, and that's probably my fault. I tend to perpetuate that, but I think that it's specifically for this session today a really great way to describe the journey of drug development, getting something from a great idea to an actual treatment that we can use to help patients in any disease community. So I started running half marathons in 2009, not long after my sister Taylor ran a 5K after losing her vision to Batten disease. And I wasn't very good. I was a soccer player growing up. I was really good on the soccer field, but I really didn't understand how to do that 13.1 mile thing. I just didn't understand the strategy of a long race. And the way I learned to do it, the way I kind of mastered that distance was I started running those races in 3 parts. And just bear with me a minute. So if you've ever, show of hands if you've ever run a half marathon or longer. Okay, so got a lot of runners in the room. That's awesome. So the way that, I don't know how you did it, but the way that I learned was I started thinking about it in 3 parts. And it was when that starting gun goes off, so that first mile of the race and really getting into my rhythm, finding my stride. It's the middle of the race where, depending on the kind of day you're having, why the hell am I here? Why did I get out of bed at 4 a.m. this morning when I could have been at home? Why am I putting myself through all of this pain and suffering and getting Gatorade thrown on me at water stops? And there's a game on at home and I could just be sitting on the sofa. And then at the end, when maybe you see the finish line, but you're not there yet, and you really have to dig deep, and your ankles and your calves and everything in you is screaming at you and say, you're crazy, why are you doing this to me? And I think that those are really the 3 most important parts of the race. And drug development is kind of the same way. I think that you have to be focused from the very beginning. You have to have drive. But then you have to be willing to stay awake for the middle. You have to be focused. You can't lose faith. You can't lose hope. You can't lose that drive. And then you got to know how to save enough juice for the very end. And this last picture on the right here is actually of Steve and me. I'm running in all 50 states, and we did Alabama together because Steve is an Auburn grad, and he couldn't wait to run on the field at the stadium in Auburn. And I don't think either one of us had the best race that day. We were hurting a little bit,
terrible,
but
yeah.
it was terrible. But when we came out of the tunnel onto that field, we were able to find that juice, and we're both competitive people with ourselves and others, and I had a sprint to the finish line, and I think I might have beat him by a hair. I think we might have a picture to prove that. But
I think we have photo evidence to the contrary.
But it works for drug development, and I've been a runner almost as long as I've been trying to fight Batten disease on behalf of my little sister and others. And I think that like a long race, the race to cures is never, ever going to be easy. And if you go to any session this week and somebody tells you that it's going to be easy, I hope you come and tell me who that person was, because it's not easy. If the great stories that you hear, they don't ever talk about the long hours in the lab, which Steve could probably attest to. They don't talk about the thousands of experiments that never, ever work, that don't produce the breakthrough, that produce the wrong answer, even. Because in science, yes, I think that there are miracles. I'm a hopeful person, and I believe in miracles, but miracles don't just happen. I think miracles happen because people do believe in them, and then also because they're willing to put the work in to make those miracles happen. And I think that's, I think a lot of people here this week are living proof of that. So I think if you want to get to the finish line, and we'll talk about what the finish line actually means in a minute, you have to accept before you do anything else that it's not going to be easy. You may have some days that feel great, but it's never going to feel easy as a whole. You're going to feel winded. You're going to get injured. I've been injured too, a lot. You're going to crave those water stops more than the miles in between. And people who are faster are going to pass you, and it's going to suck, and it's going to piss you off. But then you're also going to pass the runners who don't want it as much as you do. And I think that's the really important part. You have to go all out from the start, and you have to want it every step of the way. I think the good news, bringing this back to rare disease specifically, is that, and Steve and Sharon have both alluded to this in our presentation, is that times have changed since my sister's diagnosis. Taylor was diagnosed with CLN1 disease in 2006 on a hot summer day, and I really feel like it was the dark ages of rare disease and Batten disease in general, and I feel like we've had a renaissance. I mean, global genes didn't exist when Taylor was diagnosed. I mean, think how the world has changed if you've been in the rare disease fight since then. It's incredible, and I'm really, really excited about it. There are companies that are committed to developing rare disease treatments, as Steve mentioned earlier. I mean, why the heck would a clinical stage company care about investing in something where they may only have 100 customers in the whole world? And that's really powerful. And I think that advocates in this room today take credit because you've made that happen. I always say, and this is the sad part for our family, that I think that if Taylor had been born 10 years later, our journey may have been different. And we may have saved her life. But instead, it's not her life, it's her legacy. I hope that you're inspired to get to work. It's just figuring out how to get started. It's figuring out what you already have to work with in your disease community. It's understanding that you don't have to raise millions of dollars necessarily. You could, and Steve could attest to this, I mean, I think you could fund a mouse model if you don't have one for maybe $100,000. It's not talking, you're not talking 6 figures, you're not talking 9 figures. You have to start figuring out those pieces that you can contribute to and get things going. What you raise may actually be less than, say, 1% of the total cost to get a drug to market. But again, thinking back to a race, that first 1% may be the most important part, because if you don't get that 1%, then nobody else is going to care enough to get started in the first place. The patient community has to be the catalyst in drug development. You have to make sure that science is strong so you can attract commercial partners. And I'll let Steve briefly talk to what some of those things are.
Yeah, well, so one big point that I want to make is, right now, I think my lab is working on about 24 different diseases. We've got 9 things moving into clinical trials right now. And every single one of those started because a parent approached us or a foundation approached us. And then we looked at the biology of it and said, does this make sense or not? And if the biology made sense, and the foundation was able to be a good partner to help us drive it forward, then we worked on it. And there are cases where the foundations have, they're a few $100,000s in, and then we've gotten commercial partners to pay when it gets to be 10 times more than that to move it into a clinical trial. And that's becoming kind of the evolving model that can work for a lot of these things. But it really starts with the families and the foundations that drive it forward. Without that, it's not going to happen.
I think we've got just a minute or two left, so I want to close this so we'll have time for questions. I don't know how many of you have been able to get a copy of David Feigenbaum's book, Chasing My Cure. It's a book that came out on September 10th. I was able to get an early copy, and I want to read a quote quickly from that book. "Fear disintegrates, doubt disorganizes, hope clears the way, pushes out the horizons, and gives us space to build structures." So what exactly does that mean? To me, I haven't asked David what he thinks it means, and he wrote it, but I think that being invincible in hope means having the courage and the will to take action, because you can't trust and wait and hope that somebody else is going to fix the problem for you. You can't assume that a human or a higher power is going to save the day. That's not real hope. And so ring this is really short, I promise, moderator. This is my book that came out last year. And this will bring us back, hopefully, to when we met Steve. "I will always remember the morning we announced our gene therapy grant as if it happened in an alternate universe. Outside the hotel where 80 people had gathered to hear our news, the world was cold and cruel and vast. But inside the banquet hall, I felt warm and safe, euphoric even. When I stood at the podium and told the crowd about my little sister and the terrible things happening to her, I felt stronger than Batten disease. I heard myself telling them she was blind and couldn't learn like other kids and suffered from seizures. I heard my own words describing how Batten disease would steal her speech and put her in a wheelchair and how I would probably outlive her, even though she was 16 years younger than me. But up on that podium, none of it felt real. And when I introduced Steve Gray, I believed in the project. I believed in the evolution of gene therapy and the world-class team and the success of the GAN research. I believe because of Steve's track record and his heart for kids with no hope of a future, and most of all because of my mother's gut feeling that he was the one. I could tell our audience believed in Steve too. He had a way of putting complex science into relatable terms, and the crowd connected with him because of his humble, down-to-earth nature. It was impossible, too, to deny the potential of the work he'd been doing for Batten disease. The enthusiasm in the room was palpable, and immediately following the program, people flooded us with hugs and handshakes and promises of support for our mission to save people like Taylor. "This was amazing," I said in the parking lot afterward. "We were the last ones from the breakfast remaining at the hotel, and some of the excitement had started to wane, but I could still feel the smile I'd had all morning in my cheeks." "It was great," Steve agreed. "I'm excited about getting back to work at the lab." He paused for a moment. "We'll have to be relentless, you know. Nothing worth having is ever easy." And I wanted to end on that because I love that word, relentless. And I may have paraphrased him a little bit. There were a couple of years between that day and when I wrote that paragraph. But relentless is such a powerful word. And I think that the reason you see the 3 little ellipses on the finish line, the slide up here, is that there are lots and lots of finish lines. You're going to cross one, but then there's going to be another and another and another. To get through that, you have to look inward. You have to take an active role in transforming your hope into reality, because that's hope in action, and that's how you can transform the world. So, thank you.
Thank you, Sharon, Steve, and Laura. That was a great message. We're going to have a few minutes for Q&A. So let me just remind you, some of you have already sent me some questions here on the app. If you prefer to send your question in on the app, I'm here, I'll receive it. In a minute, I'll invite you up if you prefer to come up to the mic. As you do ask your questions, please just make them as appropriate for everybody in the room and not specific case studies, okay? So Steve, I think this first question is for you. Don't you have to get a copy of the working gene into every cell in the body? Isn't that the only possible way modifying the germ cell?
So this is a difference between if we're talking about a cure and we're talking about treatment. So if in a perfect world, if we could correct the gene in every single cell in the body and we could do it early enough in the disease progression, yeah, that would be the definition of a cure. But the reality is we're treating a portion of cells. We're trying to target the cells that are most critical to the disease progression. So we're correcting part of the body, part of organs, which, I mean, it's been shown across multiple diseases. I mean, that can end up being really transformative in terms of the benefit. But, and then when you're talking about germ cells, that's ethically, that's something that nobody's really touching because it's basically, you're specifically not trying to do that. You're not trying, so if you treat somebody with gene therapy, they can still pass that gene on to their children.
Thank you. Laura, earlier you spoke about your international work, or maybe it was Sharon. Maybe you can answer this. How do treatments developed here translate to patients worldwide in other countries? Can families from around the world enter into clinical trials in the U.S. receiving treatments developed here, etc.?
I think they said to me, but you could take that one.
Oh, it's case-by-case basis. I mean, many rare diseases, the patients are, there's so few patients and they're scattered all around the world that clinical trials, in order to bring in enough patients to actually test the drug, they're brought in from, they come in from all over the world. Our GAN trial, probably half the patients are international and half are US.
Laura, this is specifically for you. What is the name of your book?
It's Run to the Light. So, and it came out serendipitously just 5 weeks after Taylor passed away last fall. And on the same weekend that I ran the half marathon blindfolded for again and got to do that with Steve. So.
Questions from the room? Audience in the room? Come on up.
Thank you so much for the presentation. It was wonderful to see that, and Dr. Gray, it's amazing that you have this gene therapy platform that you can make that for so many different diseases. My question, as someone who works in the industry, is number one, how do we find you guys so that we can find those de-risk assets and hopefully take them to the clinic? Or to the next phase, I should say, to phase 2, phase 3 registrational studies. Then the second question that I have is, me personally, it's very hard to come up to you and say, hey, I'm from a pharma company and I'd like to take this forward because I feel like I'm making money off of patients. And I know obviously that's what we need. We need the pharma companies to take this to the market, but any suggestions on how to?
So, well, maybe not specifically answering that question, but I think on the theme of your question, one of the things, I'm never going to say big bad pharma, and we shouldn't do that. We shouldn't be demonizing people because it's a capitalist system, right? Because ultimately things are expensive, and the money has to come from somewhere. And when we're thinking, most of us in this room are thinking, what can I do just to get into a phase 1 trial? What can I do to get there? But the reality is, we don't all want to treat 10 patients in a clinical trial and have a few winners and have a lot of losers. We want these things to be successful and to turn into approved drugs. And when you start talking about the money that's involved with that, maybe you concede a few $100,000 to treat a mouse, and then it's times 10 to do the initial human testing, and then it's times 10 again or more to become an approved drug. And you're funding to seed treating a mouse is like 1% or less than 1% of all the dollars that have to go into this treatment being available to everybody that needs it. So, and this isn't inflating costs, this is real cost. And the only, unless there's, unless you win the lottery or have some, magic money tree or something like that, the model for how to get the drug to every patient that needs it, it has to involve a company. And so a lot of, in thinking about this and in thinking about how to pay for things, you really have, if your goal is to treat your child, and that's it, okay, that's a different question. If your goal is to make sure that no parent goes through what you're going through now, then you have to have a long game of what is it going to be to have an approved drug. And you have to think early about how to set it up to, have some model for how a company can get involved to commercialize it.
I would add to what Steve is saying, and I come at this again from the perspective of somebody who fought this for a long time, hoping it would save my sister, but then ultimately it didn't. I think that if you have a rare disease, if you love somebody who has a rare disease, they have to be, my mom calls it your North, Taylor is her North Star. You have to have that emotional piece that really drives you every day, but it can't be your only end game. And I say that not, regardless of whether or not you have enough time to actually find an answer for yourself or your family member, it's also about thinking just the way the world works. I mean, you have to, I think if you have that broader perspective and how can this help more people, then you're going to be more likely to get to that goal just because of the way that the industry works and the way that drug development is set up.
And the reality is things always take longer than you think they do. I'm not telling anybody anything you don't know. But so, I mean, I've had a few things, like with GAN, where there's patients that I knew that got treated in the clinical trial, and now, before their book was written, now their pages are blank. But I also know a number of GAN kids that have passed away, that despite our best efforts, we weren't fast enough. And that's reality. I mean, I think that we work with the hope of treating the kids that we have today, but we ultimately, we want to make sure that other families and other parents don't go through what you're going through.
Thank you very much.
So a lot of the rare groups tend to think of gene therapy as the panacea. And Steve, you've been very careful to point out the difference between treatment, which is really the goal, and cures, which is really the, knocking it out of the park. My question relates to sort of balance, because things always sort of swing in trends, and the pendulum is now swinging towards gene therapy, and everybody is thinking this is the panacea. 20 years ago, the panacea was small molecules, and you couldn't get gene therapy funded to save your lives. How do you think about the balance? Because the old methods work as well as the new won't work for everything. How do you think about balancing those?
Yeah, I mean, ultimately, you've got to understand the details of your disease and what's practical, and that's a hard thing. That's easy to say, it's hard to do. I think having good scientific advisors, a variety of different scientific advisors that you trust, to look hard at the science and see if something makes sense or not. Don't just go with the glittery, glamoury thing. Actually see what applies to your disease and what makes sense. And with GAN, I mean, we started the clinical trial, we've dosed 11 kids. We're still in the lab, we're researching to try to make it better. And our approach is, the perfect can be the enemy of the good. If you sit there and try to make the perfect treatment, then it's going to take forever. It's probably never going to happen. But if you stop at some point and say, this is good enough, I think this can help the kids, at least buy them some time, at least make their lives better, let's take it forward. And then let's keep on working on the next finish line.
I think with rare disease, so many of us start with nothing, that a phase 1 clinical trial feels like that ivory tower. Like we've arrived, we've made it, we're done. And that's, while incredibly exciting, I think it's dangerous thinking. You know, you think about common diseases and conditions. I mean, we didn't stop studying high cholesterol just because we got a cholesterol drug. And we have to keep going back to the lab and modifying and fine-tuning and making things better and finding new things.
I think we have time for one more question, and I have Blake standing here beside me, so I'm going to allow Blake to ask that question, and then we'll close, okay?
That's a lot of pressure, and I hope I can articulate it. But in talking about economies of scale and talking about all the communities and all the drugs in the pipeline for the gene therapy trials, AAV is fairly common. There's a lot of exposure. How, from the clinical trials standpoint, how can we recruit people and screen them to see if they've been exposed to it? I mean, because we're having to fill all these trials, and I feel like that's especially in the Duchenne space, is finding these people that haven't been exposed to it. Is there a process that can happen early on to help fill those trials and that sort of thing? I wanted to get your take on that.
Yeah, so just if everybody doesn't understand this, when you're talking about using viral vectors to deliver diseases, these are viruses that are natural in the human population, most of them. And so like a third of us in this room have antibodies against AAV9. So if we were to treat you IV with an AAV9 vector, your body would just completely neutralize it and reject it. So you can't get that treatment. Now there are other approaches of injecting the virus different ways that would avoid those antibodies. The whole field is working on this and trying to figure out, are there ways to, you know, neutralize the antibodies and allow dosing those patients. Everybody recognizes this is the problem. There's a lot of smart people that are trying to figure it out, but it's just right now today, we're still trying to figure it out. In terms of advice for how to, as a parent, like how to deal with that or how to cope with it, I don't have a lot for you.
Sharon, Steve, Laura, thank you. Let's give them a round of applause, please. At this time, we're going to ask that you jump on the mobile app, fill out the survey for Laura, Steve, or for this session, let's say. And we're going to make our way to lunch. Lunch today is sponsored by Pfizer. So if you run into somebody from Pfizer, thank them. And on your way out the door, there is a resource table. Pick up those resources as appropriate for you.